Hello and welcome to my video. Alida this side. Okay, today we will start biotechnology and its applications. Okay, so one important thing to say before we start the chapter. I will omit several portions, and I will only tell you the important portions from where the questions are coming in the previous years. Okay, so let's do this. Okay, all right. So we are going to start from the applications of biotechnology. uh this part. From this to this we are going to do. Yeah. Okay, let's start. The applications of biotechnology include therapeutics, diagnostics, generally modified crops for agriculture, processed food, bioremediation, waste management, and energy production. Three critical research areas of biotechnology are providing the best catalyst in the form of improved organism, usually a microbe or pure enzyme. Okay. Second, creating optimal conditions through engineering for a catalyst to act, and downstream processing technologies to purify the protein or organic compound. Okay, so applications of biotechnology. What are the various applications of biotechnology? Applications of biotechnology are seen in therapeutics, diagnostics, generally modified crops or GMOs, crops for processed food, bioremediation, waste treatment, and energy production. In all this field, biotechnology has its respective applications. The critical three critical research areas of biotechnology, they are, as I told you, to provide the best catalyst in the form of improved organism, usually a microbe or pure enzyme. Okay. Second is creating optimal conditions through engineering for a catalyst to act. Okay. And third is downstream processing technologies to purify the protein or organic compound. Right. Nothing much to explain in here. Let's move to the next part, that is biotechnological applications in agriculture. Okay, so we already studied that applications of biotechnologies in genetically modified crops, processed food in agriculture. Okay, so we are going to study these in detail. Biotechnological applications in agriculture. Three options that can be thought for increasing food production: agrochemical based agriculture, then um, organic agriculture, and genetically engineered crop based agriculture. So, three options that can be thought for increasing the food production include genetically modified crops, crop based agriculture, organic agriculture, and agrochemical based agriculture. Nothing much to explain in here too. All just you need to memorize them. Okay. Now let's study Green Revolution. Okay, Green Revolution succeeded in tripling the food supply, but yet it was not enough to feed the growing human population. Increased yields have partly been due to the use of improved crop varieties, but mainly due to the use of better management practices and use of agrochemicals, fertilizers, and pesticides. However, for farmers in the developing world, agrochemicals were too much expensive, and further increases in yield with existing varieties that are not possible due to conventional breeding. Is there any alternative path that our understanding of genetics can show, so that farmers may obtain maximum yield from their fields? Okay, so this total part, you don't need to um, actually memorize this, but uh, obviously the next part you need to memorize. Okay, there are several things that you need to memorize. And okay, so from here we are going to study about tissue culture. Okay, as traditional breeding techniques failed to keep pace with demand and to provide sufficiently fast and efficient systems for crop improvement, another technology called tissue culture got developed. What does tissue culture mean? It was learned by scientists during 1950s that whole plants could be regenerated from explants. What are explants? That is any part of a plant taken out and grown in a test tube under sterile conditions in a special nutrient media. This capacity to generate a whole plant from any cell or explant is called totipotency. So what happened? Uh, what is a tissue culture technology? In tissue culture technology, what happened? Scientists understood that plants could be regenerated from explants. What are explants? Explants are the any part of a plant that is totipotent. What is totipotent? What does totipotent mean? Totipotent means it has the potential to produce one whole plant from that small part of a plant. That is totipotent. So animal cells are totipotent because we start our life from a single cell, that is zygote, and then we grow into a whole organism, so we are totipotent, right? So this is totipotency. Okay, the capacity to generate a whole plant from any cell or explant is called totipotency. You will learn how to accomplish this in higher classes. It is important to stress here that the nutrient medium, of course, there must be nutrient medium, because that single cell will obtain nutrients and then it will grow. So of course there is a need of sufficient nutrients, okay, for growth and development, since the whole thing is being prepared in vitro artificially. So of course there is a need of sufficient nutrients, and as a nutrient media, the the number of nutrients or what kind of nutrients must be there also matters for the growth of the plant. Okay, so it's not a very simple process. Of course it's a very tough process, right? So you will learn how to accomplish this in higher classes. It is important to stress here that the nutrient media must provide a carbon source such as sucrose, and also inorganic salts, vitamins, amino acids, and growth regulators like auxins, cytokinins, etc. Of course you've heard about auxins, cytokinins, gibberellins, right, ascorbic acid. All are like plant growth promoters, inhibitors. All of these are all phytohormones. You've read about this, right? So there is a need of all these, like carbon rich sucrose. There is a need of these phytohormones. There is a need of vitamins, minerals, and organic salts, amino acids for the growth and development of the explant. By application of these methods, it is possible to achieve propagation of a large number of plants in very short duration. So it is what? It is continuous culture. What are we doing? We are preparing very large yield of that plant. Okay, in case of tissue culture, from that small explant, we are creating many plants, a large population of plants, a large yield of plants. So this is continuous culture. Okay, we have read about two kinds of culture, batch culture and tissue culture, if you remember in the last video. So whatever, let's see. Okay, so this is a continuous culture, a kind of continuous culture. So by application of these methods, it is possible to achieve propagation of a large number of plants in a very short duration. So this is not something the traditional hybridizations technique could have done. This is a product of biotechnology, right? This method of producing thousands of plants through tissue culture is called micropropagation, right? It is called micropropagation. Each of these plants will be genetically identical to the original plant from which they are grown. That is, they are soma clones. Obviously, these all these plants that are grown through tissue culture or through micropropagation, they're all are identical to each other. Okay, and thus they are called soma clones. These terms are very important. Explants, tissue culture, totipotency, then soma clones, micropropagation. You may be asked, what is tissue culture? What are explants? What do you mean by totipotency? What is micropropagation? What are soma clones? What do you mean by soma clones? And another thing that I want to tell you, that is you know callus. So while this explant or this growing tissue divides, it divides and forms an unorganized mass of cell, and that is called callus. Okay, you have learned it in your younger classes if you remember. Okay, it is not needed in here actually. Whatever, let's see. So maclones, many important food plants, like food plants means plants from which we get our food, right? So food plants like tomato, banana, apple, etc. have been produced on commercial scale using this method. So remember the examples: tomato, banana, and apple, etc. have been produced on a commercial scale using this method. Using what method? Using micropropagation. Hmm. Okay, so try whatever, leave this line. Another important application of this method is the recovery of healthy plants from diseased plants. Even if a plant is infected with a virus, the meristem, the apical, and the axillary is free of virus. Hence, one can remove the meristem and grow it in vitro to obtain virus free plants. Scientists have succeeded in culturing meristems of banana, sugarcane, potato, etc. So, here also the examples are very much important. So, with the help of micropropagation, tissue culture, we have obtained various food plants like apple, banana, and tomato. And another important application of biotechnology or micropropagation is that if suppose any plant is diseased, so it will die. But we can, with the help of totipotency, we can now extract the apical or the axillary meristem of the plant, because explant can be any small part of the plant, right? Either I mean any small part of the plant. So we can extract the axillary or the apical meristem of the plant, because even if a plant is diseased, then those two parts will be free of the disease, like free of the virus or bacterial infection, and we can extract them. We can take the explant, we can uh, um, give it a nutrient rich medium, and after a few days it will give us a large population of plants. Okay, through the micropropagation, through the technique of micropropagation. Okay, so this whole process is taking place in vitro, and of course the plants which have been produced, they are virus free plants, they are infection free plants. Scientists have isolated single cells from plants, and after digesting their cell wall, have been able to isolate naked protoplasts surrounded by plasma membranes. Isolated protoplasts from two different varieties of plants, each having a desirable character, can be fused to get hybrid protoplasts, which can be further grown to get a new plant. These hybrids are called somatic hybrids. Well, the process is called somatic hybridization. Imagine a situation when a protoplast of tomato is fused with that of potato, and they are grown to form new hybrid plants combining potato and tomato characteristics. Well, this has been achieved, resulting in the formation of pomato. Unfortunately, this plant does not have all the desired combination of characteristics for its commercial utilization. So, what happened? Another way in which these another important application of biotechnology in agriculture is that uh, what happened, scientists isolated single cells, and from them scientists even isolated the naked protoplasts of the cells just surrounded by the plasma membrane. So uh, like this, they took two varieties of plants and they isolated their naked protoplasts. They just the part that is surrounded with the protoplasm and plasma membranes. Okay, and they isolated the protoplasts from two varieties of plants, two different varieties of plants, and each one had a desirable variety that the scientists are wanting from them. And what they are doing is they are fused, these two protoplasts, they are fused to get a hybrid protoplast, and this hybrid protoplast will be further grown to give us our hybrid plant, hybrid new plant. So these hybrids are called somatic hybrids, and the process is called somatic hybridization. So if suppose protoplast of a potato cell is combined with the protoplast, is fused with the protoplast of a tomato cell, then we will get a fused protoplasm which contains characters of both potato and tomato, and the resultant plant is pomato. And an interesting fact about this tomato plant was that it had a potato under the ground and tomato hanging from the branches, okay, above the ground. So that was the characteristics of the plant. So from one plant we are getting two kinds of food. Okay, so this is the you know the application of biotechnology, somatic hybridization. So we read there are three important applications of biotechnology that we read. That is micropropagation, used in two ways. First is used as an explant is used, and a large variety and a large yield of plants are obtained from a single explant. Another is that if suppose a plant is diseased, then we can produce healthy plants from apical meristem or axillary meristem which remain disease free commonly. Okay, and third is hybrids. We can get somatic hybrids, okay, or soma clones from a process which is known as somatic hybridization, right? Okay, so now we will read about the next part. Hmm, that is uh, plants, bacteria, fungi, and animals whose genes have been altered by manipulation are called genetically modified organisms. So, what are genetically modified organisms? Plants, bacteria, or fungi, or animals whose genes have been altered by manipulation are called genetically modified organisms, GMOs. Genes can also be altered through mutation, but that is not genetically modified organisms, right? If they are altered by manipulation, then they are called genetically modified organisms. Genetically modified plants have been used in many ways. So, this part is important again. Genetic modification has made crops more tolerant to abiotic stresses, cold, drought, salt, heat, reduced reliance on chemical pesticide, pest resistant crops, helped to reduce post harvest losses, increased efficiency of mineral usage by plants. Okay, this prevents early exhaustion of fertility of the fertility of the soil, enhanced the nutritional value of food. Its example, golden rice, that is vitamin A enriched rice. So what are the applications of biotechnology or what are the advantages of genetically modified crops? There are total 5 points. Okay, it has biotechnology or genetically modified crops have become pest resistant. Okay, now they can actually withstand pest attack since they are made pest resistant crops. Number two is made the crop um, tolerant to abiotic stresses, like if there is cold, there is a drought, okay, humidity, salt, heat, whatever, any kind of abiotic stress. Now the plant can withstand all those abiotic stresses because of genetic modification. Next, helped to reduce post harvest losses. Now the post harvest losses has been reduced. Number 4 is increased efficiency of mineral usage by plants. This way, the means when a kind of crop is okay. So when indigo, let's take an example. If indigo is cultivated on a field, that plot of field loses its fertility. Okay, and we cannot cultivate any other crop in that soil. So now, with the help of genetic modification, we can do what? We have increased the efficiency of mineral usage by plants, and this way the fertility of the soil is retained. Okay, and the fertility of the soil is not exhausted early. Next is enhanced nutritional value of crops. Golden rice has been prepared, and golden rice is rich in vitamin A. In addition to these cases, GM has been used to create tailor-made plants to supply alternative resources to industries in the form of starches, fuels, pharmaceuticals. Not important. Okay. Next, some of the applications of biotechnology in um, detail are the production of pest-resistant crop. Obviously, crops are made more abiotic, stress resistant, pest resistance, reduced post harvest losses. Number 4 is increased efficiency of mineral usage by plants, enhanced nutritional value of this, the application of biotechnology in making pest resistant crop. We are going to read that. Okay, pest resistant plants, which could decrease the amount of pesticide used. If we can make the crops pest resistant, then we don't need any additional pesticide or insecticide. Hmm? That will, uh, okay, um, okay, amount of pesticide used. Bt toxin is produced by a bacterium called Bacillus thuringiensis. Spelling is very important. Bt for short. Bt toxin gene has been cloned from the bacteria and had been expressed in plants to provide resistance to insects without the need for insecticides. In effect, created a, a biopesticide. Examples are Bt cotton, Bt corn, rice, tomato, potato, and soybean. So, Bt toxin is produced by a bacteria which is called Bacillus thuringiensis, and from this bacteria, this Bt toxin gene has been isolated and has been expressed in plants, and we have seen that this Bt toxin has made the plant pest resistant, and now there is no more for pesticide or insecticide in certain cases for the plants. Okay, so how do we make Bt cotton first? So Bt cotton, some strains of Bacillus thuringiensis produce proteins that kill certain insects such as lepidopterans, tobacco budworms, armyworms, coleopterans, beetles, and dipterans, flies, and mosquitoes. A mnemonic that I want to suggest for this is LCD. Just remember LCD. L for lepidopterans, uh, C for coleopterans, and D for dipterans. Okay, and for L, I mean lepidopterans, it is BA. Okay, B for budworm and A for armyworm. B for tobacco budworm and A for armyworm. For coleopterans, it is B, for bee, it means beetle, and um, for dipterans it is FM. Okay, FM, full marks. So F means flies and M means mosquitoes. uh Now what happens? um Form, okay, um thuringiensis, bacillus thuringiensis forms um protein crystals during a particular phase of their growth. These crystals contain toxic insecticidal proteins. Okay. um why does this toxin does not kill the bacillus itself? However, the beta toxin, a protein, exists as inactive protoxins, but once an insect ingests the inactive toxin, it is converted into an active form of toxin due to the alkaline pH of the gut, which solubilize the crystals. The activated toxin binds to the surface of the midgut epithelial cells and creates pores that cause cell swelling and lysis and eventually cause death of the insect. Okay, so what happens in case of bt cotton? So some cells of bacillus thuringiensis, they produce certain proteins which kill certain insects like uh, LCD, that is Lepidopterans, Coleopterans, and Dipterans. Lepidopterans means BA, that is tobacco budworm and armyworm. For Coleopterans, it's beetles. For Dipterans, it's FM, that is flies and mosquitoes. Um, these are the insects that are killed by BG toxin. So what happens? Bacillus thuringiensis, it forms certain protein crystals during a particular phase of its growth. But this does not kill the bacillus. I mean, Bacillus thuringiensis itself, because it is retained in its body in the form of inactive protoxins. But when an insect ingests it, when that inactive protoxin crystals come in contact with the alkaline pH of the gut of the organism, at that time the crystals solubilize, and this activated toxin lines the midgut, the surface of the midgut, the cells of the epithelial cells of the midgut of the insect, and it creates a cell swelling and lysis, and eventually um, makes pores on the gut of the insect and leads to the death of the insect. Specific Bt toxin genes were isolated from Bacillus thuringiensis and incorporated into several crop plants such as cotton. The choice of genes depend upon the crop and the targeted pest. As most Bt toxins are um, insect group specific, the toxin is coded by a gene, uh, cry um, okay, cry-ISC, named cry. There are a number of them. For example, the proteins encoded by genes cry-ISC are cry1AC and cry2AB, control the cotton bollworm, cotton bollworms. That of cry-AB controls corn borer. I'm so sorry, that was okay. um I'm sorry. Okay, let's revise this another time. Specific Bt toxins were toxin genes were isolated from Bacillus thuringiensis and incorporated into several crop plants such as cotton. Okay, it was incorporated into cotton and also into corn, and also into corn. Okay, so the choice of the genes which will be incorporated depends upon the plant and also upon the targeted pest of the plant, right? So most bt toxins are insect group specific. The toxin is coded by a gene. So this bt toxin is coded by a gene which is cry1AC, or named cry. There are a number of them. For example, the proteins encoded by the genes CRY1AC and CRY2AB control cotton bollworm, and that of CRY1AB control cotton borer. So CRY2AB and CRY1AC control cotton bollworm, and um, CRY2AB control our what is called corn borer, a targeted pest of the corn plant. Okay. Okay, so in case of pest resistant crop, another important example is of the nematodes. Okay, nematode, it's a parasite that parasites on a wide variety of plants. Okay, and the major example is that of meloidogyne incognita. The spelling will be in NCRT. The spelling is given as meloidogyne incognitia, but it should be meloidogyne incognita, not tia. So actually the spelling will be, I'm writing it here, sorry, wait, okay, the spelling will be meloidogyne incognita. You can search it on internet. Okay, so cut the I of incognitia and cut the E of meloidogyne. Okay, so meloidogyne incognita infects the roots of tobacco plants. So what happened? RNA, the conception of RNA interference was used here. What is RNA interference? In eukaryotes, in all the eukaryotes, uh, a, a kind of phenomena happens due to as a method of cellular defense. So what happens? Double-stranded RNA goes and binds to the mRNA, to the complementary mRNA, and thus silences the mRNA. Okay, so Agrobacterium tumefaciens is a good vector. We studied it in the previous chapter. So using Agrobacterium vectors, what was done? Nematode specific gene was incorporated in the host plant, that is the tobacco plant. And what happened when the DNA was introduced? It produced both sense and nonsense RNA, and the sense and nonsense RNA, they are complementary to each other, and thus they form double-stranded RNA, and this double-stranded RNA silenced the specific mRNA of the nematode Meloidogyne incognita. And what was the result? The result was that the nematode Meloidogyne incognita could not survive. Okay, so let's read. Several nematodes parasite a wide variety of plants and animals, including human beings. Nematode Meloidogyne incognita infects the roots of tobacco plant and causes a great reduction in yield. A novel strategy was adopted to prevent this infestation, which was based on the process of RNA interference. RNAi. RNAi takes place in all eukaryotic organisms as a method of cellular defense. In this method, this method involves silencing of a specific mRNA due to a complementary dsRNA. The molecule the dsRNA that binds to and prevents translation of the mRNA, thus silences mRNA. Okay. So, the source of this complementary RNA could be from an infection by virus having RNA genomes or mobile genetic elements, transposons, that replicate via an RNA intermediate. Using agrobacterium vectors, nematode-specific genes were introduced into the host plant. The introduction of DNA was such that it produced both sense and nonsense and antisense. Sorry, I said nonsense, right? Antisense. Antisense sense and antisense RNA in the host cells. These two RNAs, being complementary to each other, formed a double-stranded RNA that initiated RNAi, that is RNA interference, and thus silenced the specific nematode. The specific mRNA of the nematode. The consequence was that the parasite could not survive in a transgenic host expressing specific interfering RNA. The transgenic plant therefore got itself protected from the parasite. And this is how biotechnological application was seen in agriculture. Next, we have biotechnological applications in medicines, but I will discuss it in my next video. Till then, thank you.